let me move this movie here. Okay. Let's continue from the previous. I think we will discuss about implantable cardioverter defibrillator. I think we already discussed a little bit of this before. So here you see, I strongly recommend this ICD implantable cardioverter defibrillator surgery. It takes 10 minutes. It shows how things work in surgical place so that you guys can understand how things work. Here, this is a pacemaker. And also I want to show, let's see. So my face might be better to be here. Okay, see, so this is a defibrillator, and this will be implanted into the patient. We make a small surgical incision and make a pocket, and that lead goes through this vein leading to heart. And these electrodes can go at the right atrium or at the ventricles to give out the electrical pulse, which makes the irregular heart rhythm regularly. So that's a pacemaker, but very likewise, here's the defibrillator. You see how big and thick it is. So this one, as I said, is ICD, implantable defibrillator, which you put it there. And you see this one has a number of holes in it here. So it has a multiple leads. And the reason is you have to listen to the heart. So that's ICD detect, so that when the heart shows arrhythmia and a sign of fibrillation, and fibrillation is a fine irregular movement, so that we call either ventricular fibrillation or atrial fibrillation. So atrium versus ventricle is more important. So ventricular fibrillation means the heart cannot pump at all, and this is an emergency. And when ICD detects it, it will give out shock, a large amount of current to recover this abnormal cardiac rhythm. So it literally can save life for those who have this tendency of ventricular fibrillation. Here, fibrillation is an uncoordinated contraction. So that means, you know, for properly heart to pump, it has to do this rhythmical and sequential coordinated contraction. But when it goes like this, you can't pump. So that status is called fibrillation. So how does this ICD device works, and what does it do? And as you see, so one lead can be listening, but the other lead can be actually giving out the pulse. So you can see here a pulse generator over here. Okay, so here implantable cardioverter defibrillator. Why this name is we call it cardioversion. So you convert this irregular state into a normal state. Okay, so now from today we'll discuss about biomedical imaging. The first, so when we call imaging, we can consider two aspects. So one is anatomical aspect, and the other is a functional aspect. So we can call either you watch, let's say you are making friends with someone else, or you are making friends, and how do you know some other person? First, you want to see their appearance, okay, like their body, their shape. So this is called morphology, or we can call it anatomy. But the shape itself doesn't tell much about the friend who can be potentially harmful to me or beneficial to me. And how do you know that? And only after some time you make friends and go together, eat together, go out together, and talk about each other with conversation, you start to realize and understand the characteristic of the person. And that person is really good person only after you interact with them. That's a character, and we call it as a function. So imaging is like understanding the entity, in this case, biomedical engineering, the body parts or tissue, cellular level, we want to see that morphology first. And then ultimately you want to understand their function. But these morphology and functions are closely linked. And that morphology part, we start to talk about anatomy. So anatomical imaging, there are a number of very interesting and important imaging modalities have been developed. So first is MRI. And here I want to share with my own brain. You know, this is the first time I look at inside of my brain. Let me play. If you don't see, please let me know. So you see this is a planar view from bottom to top. You see my nose and my brain and my eyeball showing up here. So this happened in 2010. I volunteered to take MRI imaging. Actually, it was MR spectroscopy, but for that, we have to take this anatomical imaging first. So what do I learn from here? So very interesting, as you can see, the guy who took the image told me that, hey, you have problem in the nose. If you look at this nose, there's a little bit of a curved one. So I got surprised. Hey, how do you know that I've been suffering from my nose problem for decades? And he said, like, showing this you see this curved structure. And I was a little bit concerned that, hey, my nose maybe it's curved, but in fact, everyone is like this. Okay, there's not many one who has like real straight. So that's kind of interesting finding from my own anatomy. And this is a little different word for perception. That in fact we are not that perfectly symmetric, but our brain interpret when we see someone else's face on the photo, we interpret as a symmetric. But that's a little different issue. But MRI, as you can see, it gives a exquisite soft tissue contrast. This cannot be shown this clearly with an X-ray. And so that's a little difference between MRI and X-ray, okay? So, and the second one here is called X-ray CT. And this is X-ray CT, and CT stands for computed tomography. And that allows us to make a three-dimensional reconstruction. But typically when we say CT, you remember when you go a health checkup, you raise your arm and you take one shot of X-ray CT, that's called a chest X-rays. And this allows the clinicians to look at something's heart and the lungs. You cannot see these diffuse ones very well, but X-ray is usually a 2D image, and this allows us to detect, let's say, if the COVID, a coronavirus infected patient having a lung problem may be detected from X-ray CT. And sorry, it's not CT, X-ray. And you see this L, and this means a left side. So you are seeing the patient, like the patient is showing approaching to you, the face to you. Okay, and then CT case, you can see this is like real 3D, very nice anatomical structure, because CT is basically taking multiple angle 2 shots and reconstruct as a 3-dimensional. So here, MRI measures the response of the atomic nuclei of body tissues to high frequency. Oh, let me see. I think I have to ask a question to you guys. 경철, 민경철.
Yep.
Can you guess this?
Radio wave.
Exactly. Good, good. Okay. And X-ray produces image of internal composition of the body by passing through it and being 8 to different degrees by different material. What it means is X-ray is a very short wave, high energy photon. But when it passes through biological tissue, some part of it is being observed, especially a strong, more dense part, such as bone. So that's why you see bone is much lighter. And in general, X-ray do not show this nice soft tissue contrast because their densities are not that different, especially like this. So X-ray is being observed different degrees by different material. So this is something you want to know, like what's the contrast mechanism of this biomedical imaging. And X-ray CT scan, X-ray CT scan is medical tomographic imaging. Tomo means cut, so it's not real cut, but you have this cut through images and different angle, and using computer processing of series of two-dimensional X-ray images taken around in a single axis of rotation. So X-ray CT, you will have the X-rays moving around, taking picture, 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 picture in different angles so that it contains all 3D information and you reconstruct to create something like this. Okay, so let's move on. And so I want to show you about this. So the next is really about the functional imaging or metabolic imaging. So as I said, when we try to understand some body or someone, some human being or disease, ultimately, the functional limitation or functional disability is meaning a disease, right? So for this, we developed the MRI, which is originally anatomical imaging, but by taking multiple MRI shots while changing the status, the functional status, we actually created a new method called a functional MRI. So, for example, this is a baseline is a morphological anatomical imaging of MRI, but you realize that there are like this bright spots over here. So what does this mean? We actually overlaid by taking multiple MRI images and reveal the underlying neural activities. So that we call functional MRI. So how does this work? In fact, functional MRI do not directly see neural activity. It do not see the neural activity directly, but it's indirect. Why? This signal is coming from so-called T2 MRI, and T2 is related to the edema or a watery content, and actually it's related to deoxygenated blood. So let's say if you have a neural, if you think about something and that activates this left part of the hemisphere, and you take MRI shot, first shot, second shot, and keep taking, and then you subtract them, and this content is in fact a changes of a blood volume and blood content, and that subtraction gives out this differential signals, and that is fMRI. And now we consider this as a very good tool, in a living body without any harm, and reveal a place where these activities show. So MRI measures brain activity due to changing neural activity via mechanism called, we call blood oxygen level dependent contrast, because this contrast is coming from hemodynamic change. Hemodynamics means heme means blood, dynamics, so blood flow changes. So that it's very, very famous. We call it as a bold signal. Now I want to talk about positron emission tomography. So this we call a PET imaging. And yeah, the problem of MRI itself, just morphological one. Let's say your friend got a cancer and have been treated, but MRI shows, let's say there's a liver cancer and there's a big tumor, and then you got treated with chemotherapy and it works very well. And then, but chemotherapy is very painful, and you take MRI. MRI shows this big chunk left. However, in reality, what happens if the drug works well and this tumor is already died? So it's non-viable, but it shows in MRI. Clinicians can misled to keep giving you drug, even if the tumor has already died. So that's not a good thing. So that's where we need this functional imaging, and it shows that there's a tumor, but there's no activity, nothing. So then you don't have to, hey, this is already dead tumor, you don't have to treat. Or on the other case, there are patients, this one, like 3D PET image, who has a liver metastasis. So meaning that there's tumor lodging and growing in the liver, and you want to see the stage of the patient, whether the patient's, what's the status of the tumor, and you realize by using PET. PET is, the tumor has a characteristic to utilize glucose as a source of energy. And we have some other organs, such as you see this brain, you see bright spot in brain. Brain also utilize glucose. It's so good for brain imaging, functional imaging. And you realize these small spots surrounding. And this may implicate the tumor has metastasis and growing other places. And this reveals the status of, finding out which cannot be found easily with other imaging technique, because this one lights up for the viable tumor or metastasized tumor. And what about heart? So the PET is very important technique. However, it doesn't have a lot of high resolution. So let me show. PET is a nuclear medicine imaging, and it provides here like a 3D image of functional processes in the body. And how does the PET work? I'll explain you. So PET requires a nuclear isotope. For example, the popular one here is FDG. What it means is fluorodeoxyglucose, which means, this fluoro F is a radioisotope 18, then it will emit gamma rays. And so this is an analog of glucose. So it works like glucose. So the cells, tumor cells or brain, they actually, or heart actually, who uses the glucose, and it shows regional glucose uptake or tissue metabolic activity. Happen to be tumors have a big metabolic activity utilizing glucose. And how it works, gamma rays, this is called a annihilation. So this PET, the radioisotope gets into the cell, and this meets a nearby electron, and what happens is a positron. So you see the PET is positron. Positron hits an electron plus minus, they will annihilate, means disappearing. However, that energy become sending out 2 photons called gamma rays. But because it's starting from nothing, one gamma ray goes this way. In physics, you have heard about preservation of momentum. So momentum preservation makes one gamma ray goes this, the other should be the opposite way. So from here, and then we detect this one with detector. Then if it's at the center, you will have 0 time difference. And you know that the time difference of detection in this line at the middle is the point of where this gamma ray is coming out. So that determines the 3D location. What if I detect it, if it detects faster here than slower here, then that location is closer to here. So that detects earlier, detects later. And this is the speed of light. Speed of light is 3 times 10 to the 8 meter per second. And you can calculate, let's say PET system has about 1 meter, then it takes about nanosecond to detect this time difference. And luckily, our electronics can detect, nanosecond order. That's what you should see. This cell phone, you heard about 2.4 gigahertz. Like electronics can detect gigahertz time difference. So that's how PET can work for this. Okay, so that's, and then, different angle, but there's a caveat. Then you can ask why the resolution is not that great. If you can detect this, of course, the detector size, and more fundamentally, this gamma ray, positron is coming from FDG, this glucose, but when positron is decaying, it comes out, it has to move to meet nearby electron. And that is a random process, and that distance is about order of 1 millimeter. So that's why you cannot have a better resolution below 1 millimeter. So that's something like limitation. a fundamental limitation. And let's talk a little more about the utilities of PET. So here, permanent damage and transplant. So, you know, in our organ, using a glucose actively is our brain, which is always working. Our heart is always working. And here, this is a bladder, which happens to drain this radioisotope. So it's not really likely to be a disease. What about heart? So heart actually gives out the signal, but viable heart. So let's assume that a patient has a heart transplant, and you know, after transplant, see if this works well or not, or does this patient need heart transplant or not? So you do anatomical imaging, it shows heart, but you don't see any much. But using PET, you see there's nothing here, meaning that there's no blood flow. So this part is probably non-viable, and maybe that can say something about the function of your heart. And also here, you see this is no metabolism compared to the other part. So that is an issue. So with this patient, maybe no flow, this is that tissue. This one, maybe very low metabolism. So the patient can undergo heart surgery to recover this. So after surgery, still no flow. Oh my gosh, yeah, you know, this is that tissue. Probably you can salvage this, but after you, we'll talk about it. So there's you're suffering from heart attack or myocardial infarction due to the blood vessel is narrowed down. And let's say you have bypass surgery, which means you have another route bypassing the blocked vessel, and you realize this recovers because it shows metabolic activity, showing that, hey, we recover your heart function better. So like this, PET can be very useful. While low resolution, it provides a very valuable functional information. Okay, now I'm going to talk about a little bit of a biomedical imaging, especially optical imaging, which is actually my specialty. And I want to give you some idea of how things work. So especially this is a skin cancer. You see, this is how devastating this would be. So the important thing about skin cancer is, you know, skin is outside, okay? So nobody wants to have a big scar on your face or nose, but, you know, unfortunately, skin cancer is usually can arise exposed place because that's where you get the most UV light exposure. And so the purpose and the goal of a dermatologist to cut out your skin cancer. But when I say cancer, you know, cancer can be dangerous. You don't want to leave any like left remnant tumor. So you are having two opposing purpose. One is you have to completely remove tumor cells. The other is you want to save as much of your normal tissue. So how does dermatologist do? So you see the viable lesion on skin. So, you know, dermatologists have a rule A, B, C, D, E to determine whether it's malignant cancer or benign like others. But now AI is, you know, we are working on AI can do this up more automatically and more reliably. But once you found this, you have to do surgery. So how do you do that? So the dermatologist cut off a very thin layer. And what they do, so this is outside, you cut it off and then flip. And then watch whether there's a tumor remained or not. If there's a remaining tumor, that means the original tissue has tumors. So you flip it and then you cut through, make a section, look at under the microscope to see if there's a remnant tumor at the edge of this cutted part. And if you find at the middle something left over, then you have to go back again, another thin layer, remove. And then you find another, you go back again, only that particular location. This way, you try to minimize the cutting of your valuable skin tissue. Okay. So this was invented by a surgeon named called Mohs. So that's why we call Mohs surgery. Also, we call it Mohs microscopic micrographic surgery. And this is precise surgical technique using to treat skin cancer. During Mohs surgery, thin layers of cancer-containing skin are progressively removed and examined until only cancer-free tissue remains, which means you cut it out until you don't see it anymore. So how does this work? Hey, the patient here, removal of first tissue, it takes about 15 to 20 minutes for this operation. And then you have to send it to histology lab that you do make a section and making so-called H&E, hematoxylin and eosin, which stained for both nucleus, DNA, and the protein, okay? And then under the microscope, you examine it to see if there's a remained cancer cells. And then if some cancer remains, you go back during the time. So you repeat until it finishes, all cancer removed, then you undergo reconstruction surgery and operating room. So think about this, the patient has to wait for this time and the surgeon, dermatologist spend long hours for this. There are many other patients. So this has been a limitation. And in fact, when I was graduate student like you, I got involved in a startup company based on a patent using polarization based, how to solve this problem more fast and convenient way by using optical imaging, especially polarization property of optics. So I made a fluorescent polarization imaging device, which is quite simple in concept. So you see a laser illuminator, you send out laser light through the lens and polarizer. And this is the sample. You remember the cut sample on flip back inside out, and we shine it. And I take just picture. So it's a very simple imaging device. However, we put this sample with the analyzer, which is another polarizer. And this is actual view of this. And you see, it's very simple. And this is my first experience in company experience, but I'm the only engineer there. So I order many different types of polarizer and cameras. And you know what happens? So the sample, we shine, the polarization is the direction of electrical changes. So when the light propagates, electric field is usually perpendicular. And when it shines, this electrical field or that electrical field we call as a P and S polarization. And the concept is very simple. Just changing this polarization angle, we take pictures and then we subtract and normalize. And that's called fluorescence polarization imaging. And it actually reveals these cells, which is not very clear with the original. And what we do is at the time, Boston Medical Center. So I worked with this medical dermatologist, and when he sees it, he has an entrepreneurship mind. He look at this, oh yeah, that's the squamous cell carcinoma. And what we did is just tissue section. We just put some dye on it and then just write, and then we take picture and then process. And he said, hey, I have an intention to buy this product in $100,000. And I said, hey, no way. This is not that expensive. It's, it costs maybe $10,000 at most. But why he said this is the reality is, as you remember the previous, he said, hey, I have this clinic. I have a lab technician who has to do this H&E stain, and I have to deal with this. This guy has, salary of close to, like maybe $70,000, and I have to deal with retain this laboratory, and that's a headache. I would rather pay $100,000 this machine do the job for me and without taking much time, so that we make this Mohs surgery procedure much faster. Then it also saves the time and saves the energy for the surgeon and the doctor, and he or she can see more patients. So that's something, an example of how bioinstrumentation, in this case, biomedical optics, can help. Actually, this was a startup company, but maybe you're interested, I'll talk about it later more. Okay, let's move on to the biomolecular imaging. So, as I said, what is biomolecular imaging? which is using chemical engineering principle for design and analysis of drug delivery system and so-called tissue engineering or cellular engineering. So one example we can think of is so-called artificial tissue or skin substrate for, let's say, burn patients. So tissue engineering is the use of cells or biomaterials and biochemical factors to improve or replace biological tissue. So it requires to understand interaction of medical implants and the body. You know, we are talking about our natural body and biomaterial interaction because they sometimes touch by body or inside our body. Okay, and so biomaterials become very important in that aspect. Also, many times the drug delivery system is relevant with the biomolecular engineering. And so this aspects the emerging area of called nanobiotechnology, because you know biomolecules, the sizes we are dealing with is about down to nanometer range, which is what the proteins and this molecular site. So let's think of artificial skin. Okay, so artificial skin, this is a big issue for those who have burn patients because burn patient, that skin after like 3rd-degree, high risk of infections and all this. So we need to protect until it heals and regenerates back. So we start with the raw material called cells and biomaterials, which is scaffold. And scaffold means temporary supporting structure. So because the cells cannot organize very well by itself, so you provide this scaffold and so mixed up. And also think about how some organ can form and generate. It's not only cells, the cells are programmed, a little machine, but it interacts microscopically with the environment via these factors or biochemical. So there needs a proper cytokine. You remember cytokine storm for this COVID patients. So cyto means cell, kine means movement. Okay, what is cytokine? It's a small immunomodulating protein that usually are secreted by immune cells, and that has an effect on other cells. So it's a kind of communication tool that he tells the cells nearby, hey, you guys proliferate more because we've got injury here, okay? So, and also growth factors promotes the cell growth, and genetic manipulation is possible. And also, sometimes mechanical forces also important, like Shukla today beautifully explains, endothelial cells needs proper interaction of shear forces. Otherwise, the endothelial cell may not work properly. And all these physical chemical factors and even space and time signals are very important. And also the cells, outside of the cell, there are matrices usually composed of collagen or glycosaminoglycan, and those we call totally as extracellular matrix. So that molecules also provides information and interaction with the cell. And also, of course, how does the cell interact with others? Cell has lots of molecules on top of the surface. So all of this combined, we create engineered tissue and we make tissue substrate. How good are we? Hey, we can't produce a perfect substrate, okay? That's the limitation because our understanding is still limited, but we create at least, tissue substitute for the time to prevent infection until, let's say, one patient tissue will form a new generation with safety. So that's the status. And the next, there are many things Shukla explained, it's good. I want to talk about this carotid artery, this human sample called the atherosclerotic plague. And what is plague? Plague is this, plague is a plate on the wall, okay, like a words. From a carotid and the arterectomy specimen. So let me explain what's carotid artery. So you guys, you guys can feel arterial pulse. Everyone can do this and you can find a pulsing. So what happens? The heart, remember aorta, there's a branch to the brain. So we have to feed the brain inside and out. So there's a like Y shape. This is carotid artery. One goes to inside to feed our brain. The other goes to outside to feed our face and outside. So that's internal and external carotid, and there's a common carotid. So that's why this is Y shape. But fluid mechanical point of view, this place is where very likely to happen the fluid instability, which make the fluid flow maybe more stationary. And it happens that it's more likely to have this plaque is forming in this location. And you see this, how ugly? If this atherosclerotic plaque, when it ruptures, it can be a problem. This can go into our brain directly. It can cause ischemic stroke. Okay, so that's very dangerous condition. And used to be the doctor put stethoscope, to put here to listen. And why? Because if there's like this lesion, the flow cannot be stable. The flow will make a turbulence, okay, eddy flow. And that sound creates a sound. So clinician can listen and tell, hey, this guy has a problem here. But with the development of ultrasound, high resolution, actually, I think now we don't need that because it just shows this structure directly. It's over the years. And so what do we do of this once we found? So this is so-called end arterectomy. So what do you mean? End is inside, okay? Endo, inside, artery, and tom means cut. So inside of the artery, this guy, we cut and then take it out. So that's, sorry about a little interference, but here you see cut and take this one out. This is surgery's end arterectomy. So let me reiterate about the plaque, because we told this atherosclerotic plaque. So what's plaque? Small patch resulting from a local damage or deposition of materials. Why plaque is so important for us? Because many people die of this. Why? Especially in the heart. Let's say this vessel is a heart feeding a vessel for the heart. And this can rupture. And what does the meaning of rupture of plaque? That means inside, the material underneath the endothelial cells exposed to outside, which is blood. Then our body automatically run this cascade of blood clotting for safety, but that actually causes blockage of the vessel. And so this plaque rupture can cause so-called myocardial infarction. Clinicians say, you say MI, okay, MI. This is for normal people, it's heart attack, okay? Myo means heart, cardial means heart. So muscle of the heart is infarction. Infarction is death of the tissue due to the loss, lack of blood supply, okay? So plaque means stick, okay? So this is a blood vessel where plaque is forming. You can see there's a lot of immune cells, foam cells, and they grow to narrow down the blood vessel. And this narrowed arterial blood vessel, we call this part as atheroma, okay? And that enlarges, the arterial wall can rupture. And when it ruptures, it releases blood clots. And when blood clot forms, the blood vessel can be blocked or narrowed down. And sometimes closure means a big accident because it's complete blockage. So, okay. So let me give you an example. This is one of the most top killer in the developed country. So we call this as an atheroma, or more precisely, atheromatous plaque, because it's plaque, it's stick on the wall. It's a buildup in the wall of an artery in the arteriosclerosis. So arteriosclerosis means a hardening or stiffening, okay? So in the arteriosclerosis, we form this, you can say atheroma. Or in the other hand, the Alzheimer's disease patient have a amyloid plaque. We also call plaque. So that's extracellular deposit of amyloid beta in the Alzheimer's disease. Okay, so let me use this one to technology side, engineering side, how to cure, how to treat this. And there comes a stent technology. But for that, we have to know what happens usually in a problem in the heart. So first, you see this crown, and that's called the corona. Okay, why? You remember coronavirus shape. So this as a big vessel, as a branch, it looks like crown, and it's corona. So that's where we call the name of the heart vessel coronary artery. Okay, so as I said before, heart is feeding themselves by sending the blood vessel to feed their own muscle. And you see the shape like this round, and there are branches out. So that looks like this. That's why we name it as a coronary artery. And this is the most problematic place, which is most of heart attack happen. So that narrows down, this plaque builds up, the artery become narrowed down. And what is it like? The patients, I'll give you my example. So my mom suffered from angina. So what it means is she suffers from this symptom, then she can't walk fast because the heart is like, oof, it's difficult. And when angina happens, she described it like, hey, an elephant is, pushing my chest. So that's what it actually symptom. And how to solve this one? We create a catheter. So catheter is this one, okay? It's a medical thin tube that can be inserted into body cavity, duct, or vessel. In this case, it's a vessel. So this is a catheter to deliver so-called stent, okay? And stent is what you see. Stent is a narrow mesh, which is you originally collapsed, but once you put it here, and you use a underlying balloon to boom, to make it expand. So, and then you take it back out. The remaining expanded stent can open up this vessel, so that this is a treatment. This saves, millions of people every year. And then you think about it. So that's great, but this is a biomaterial. This is external one, and this is not even like all closed. You see, this is how much of a room between. So what happens is over time, this is regrowing through this that we call restenosis. And that become a problem because typically it takes, let's say, 5 years for this to regrow. Then you have to put another stent. That's a big problem. So people try to understand what's the mechanism of this regrowth and how do we prevent this. And the answer comes from cancer study and cancer drug. So how about putting anti-cancer drugs onto this stent to prevent? So that's something, a cool idea came out. Let me show. Stent is an artificial tubular structure. And I think I brought this stent. I don't know, is it clear? Let's see. The tiny one, and that one, and then expanded one. So that's an expanded one. Not sure whether the resolution is good enough. So this structure is inserted into natural passage or conduit in the body, in this case, blood vessel, to prevent or counteract disease-induced localized flow. Okay, so as I said, restenosis is an issue. So we have an idea of coating of the biomolecule, which is anti-cancer drug, and that drug should come out and interact very, very slowly over the years to prevent this compressed plaque to regrow. Okay, so this idea makes us to create so-called drug-eluting stent, and that actually elongate and suppress the stenosis. Let's say this stent is $1,000. This one is a $2,000, but it saves several more years for you. What will you choose? So this is, the progress of medical technology. And here, elute means removing this substance, in this case, drug, by washing with a solvent. So it's in chemistry a term, but drug is slowly releasing. So that's the technology how we make. So this is good for like biomaterials people. And let me explain another example of biomolecular engineering. So I want to tell about glider story. This is about MIT Institute professor called Bob Langer. He's a chemical engineer. He's working in the hospital and discuss with clinician about the real problem in the clinic, especially a brain cancer, brain tumor. There is a brain tumor and a malignant brain tumor called glioblastoma multiforme, called simply GBM. You know what the death rate? 100%. Nobody have survived from this. It's close to 100%. Okay, so this is a real issue and no cure. And what's the problem? This tumor is actually, when there's a tumor in the brain, if it's benign, the surgeons watch MRI and CT and make plan and cut it out, take it out, done. But the problem is the margin of the tumor. GBM has a diffuse margin, which means when you look at the microscope, tiny tumor cells are spread out and you cannot even see it. So surgeons cannot remove whole brain, right? The tumor, but there's very likely a small remnant tumor cells spread out. This is an issue. The problem of current drug is brain has blood-brain barrier. And so what it means is the chemical drug through the bloodstream cannot reach out the brain parenchyma, called the parenchyma is huge, and the business end of the brain. So it cannot get into the brain tissue very well. So he got an idea. Hey, I'm a chemical engineer. And how about after the surgery, the problem of the remnant tumor, why don't we put a drug directly over there? However, the drug has to release slowly and slowly. So you can give a local high concentration dose of the tumor right after surgery. So that's an idea. And what you have to do is to bring that technology. So after surgical resection, you create a wafer. The shape is like this. So up to 8 biodegradable discs and implant into the brain cavity left after surgery. And that disc release chemotherapy, anti-cancer drug, calmustine, which this is an alkylating agent, which basically intercalate DNA. So it prevents cells to DNA replication, okay, which continues active treatment of the tumor while the patient remains for radiotherapy. After this patient usually undergo radiation therapy. So in the mean, this will do a good job for that, okay, to suppress the tumor. So up to 8 wafers are placed in tumor resection cavity for controlled release of local dose of chemotherapy to the surrounding tissue to eliminate the residual tumor cells. And so you don't need to cross the blood-brain barrier. Okay, so that was approved by FDA in 1996 for treating after this GBM, and the only treatment for malignant glioma approved by FDA and European agency. And this is the picture, a real picture, and this is how the disc is designed. This is biodegradable, slowly releasing the drug. But I have to say, nothing is perfect. When you come up with a new idea and new treatment, you have to do this test in the clinic. And so, there's nothing perfect. So we have to also pay attention to what could be the limitation. What's the side effect of this? So adverse effect, and this is called glider. That's why I call glider story. And because this glide comes from this glioblastoma, okay? Glider wafer causes brain edema. So edema is a water deposition, or so the content of tissue water is getting higher. So it can even expand the brain sometimes, and it can be a problem for intracranial pressure. And healing abnormalities, while healing, it can also cause cerebrospinal fluid leakage. Okay, or when you do this one not correctly, there's a higher chance for intracranial infection. Cranium means brain's cut. Okay, so let me go to a little bit of a last one. for systems biology. Before that, I want to discuss about tissue culture or cell culture with you. Let me see. Let me bring some part for you guys. Because this is a Zoom meeting, I'm sorry I cannot, let's see. So first, what is, you can find out, in fact, in this corridor, you find a cell culture room or tissue culture room. And what's tissue culture? It's a growth of tissues or cells separate from the organism. Okay, you know, our body is composed of eventually down to cells or tissue. So tissue culture refers to the culture of animal cells and tissues. And sometimes we call it cell culture, which is growing and maintaining cells X under controlled condition. Let me see. Can ask question to someone. Let's see. Ijisu, can you guess what this is?
Ex vivo.
Exactly. Yes, yes, that's correct. So what I want to share with you is, so how do we grow cells and tissues outside of the body? So that condition we call ex vivo. And we say controlled condition because inside the body, it's harder to control. So what that means is we grow cells in Petri dish, okay? And this pinkish one is called a cell growth media. I brought one here, okay? You can find it, the color represents pH. So when bacteria grows, the contamination, pH change, this color turns into yellowish, then you know right away. Okay, but it contains a nutrients and proper minerals and let's say glucose, L-glutamate, and sodium pyruvate. Pyruvate. So there are biochemicals which is essential for cell growth. And this circular disc, like this, the first time when you do cell culture, you will use this one. However, this is, you know, big opening, so it's more easy to contaminate the cell. It's cheap. So because of that, when you are serious and sometimes when you're dealing with many cells, and to prevent contamination, you use this one. That shape, I don't know where it comes from. It's called T-shape and T-flask. So it's called T-flask. And the opening is much smaller that, you know, like this. Because of this, it has much less chance of contamination, while this is more expensive. But yeah, you know, you pay for what you get. And what you see here, transparent bottom, the cells, typical cells you are growing, let's say cancer cells are usually attaching on the bottom. So this is the shape of the cell under the microscope. And you don't want any bacteria or virus, you know, getting into proliferate. So that's why you need this special equipment called a laminar hood, which is basically filtered upside. So you have a no bacteria contamination from this flow. So that's what I want to say. You can grow cells, or sometimes you can grow tissue from here. So I want to discuss with you in vivo and in vitro. So how do we perform medical biomedical study? Let's say, let's have an example of attacking a cancer. Let's say I want to develop a cancer killing drugs and to test it out. So two ways of conducting biomedical experiments. One's called in vitro and the other called in vivo. And what's the meaning? It's very simple. This is all Latin. So when you write paper, do make it italic, okay? In vivo and in vitro is Latin, okay? That's why this, what it means is inside, vitro means glass. So test tube or Petri dish. Vivo means a living organism. So inside living organism, a living body or living animal, we call in vivo experiment. And you have to understand clearly what are the pros and cons of this, so that it helps you to choose which way to perform your experiment. So like this, in vitro experiment, you can have multiple of this, and you can control the temperature. You can control very well with multiple of this plate. So it's easy to control, okay? That's a good thing about in vitro. And this is much cheaper compared to animal, okay? And also it's faster. When you run your assay, hey, let's say you have this 1, 2 control anti-cancer drug with dose 1, the other anti-cancer drug, and combined drug, and you see the cancer cells are dying or not. That's your purpose. Okay, that's faster. You can, if you have an idea, you can have even weeks or month. You can write a paper. Okay, so, and also you are dealing with the cell. So for example, cancer cells are usually derived from human patients a long time ago. And so that's much less ethical issues. Okay, how does this in vivo condition account? So compared to easy to control in vivo, which means I have an animal, let's say mouse, growing tumor, and I want to treat with this drug. Hey, it's more difficult to control. And repeatability is a key for scientific research. If you report and other group cannot reproduce yours, you are doomed and nobody will believe your experiment, okay? Your reputation will drop, okay? So repetition is important, but what if this animal has already having issues of, let's say, pinworm infection or coronavirus infection, and your results maybe screw up. Okay, so in vivo, it's much more difficult to control and repeat. And animal compared to this cell, it's more expensive. And cells are faster. Animal, you have to wait them, grow them, breathe. And so it's just more time consuming. And what about ethics? You know, by using animal, you have to write down the animal protocol and ethics report and all this. It's more ethical issues. However, why do you also need to do in vivo? Because let's consider in vitro's disadvantage. While it's great, but in vitro, all microenvironment cannot be taken into account. What do I mean by that? Let's say you are studying tumor, and here in this media, it contains enough of nutrient and growth factor, and also you give an oxygen to the cell, okay? But what's the reality? The reality is why we can kill the cancer cells in the dish and sometimes even animal, why it doesn't work in the patient? Because one of the reasons, not out of many, when the tumor grows, the tumor cell has to be fed. But at the same time, you have to give the drug into the tumor. Unlike you pour this media to cell, then the drug can get in tumor directly in high concentration. So all the cancer cell can die. What about this real tumor? And there's a blood vessel, and tumor blood vessel is known to be abnormal, so it doesn't feed all the tumor. On the other hand, you give a drug, the drug cannot get into the tumor, all the cells. But even one cancer cell remains, they will proliferate. And that's a reality. And this microenvironment cannot be considered in vitro. So in vitro is great for finding mechanisms of, let's say, anti-cancer drug, but the real application requires to resolve a real problem, which is only possible with in vivo context. So for example, this one, that even if the drug is potent, what if the drug cannot be accumulated in tumor cells in high enough concentration? I say this drug delivery distribution tumor that I called pharmacokinetics. On the other hand, the tumor cells, they don't accumulate enough concentration of drug so that even the drug will not be effective to kill the tumor cells. And how the drug actually changes and kills the tumor cell, that aspect is pharmacodynamics. Okay, and hopefully this one gives why sometimes we have to undergo in vivo experiment. even though you find and you develop a new drug, which is anti-cancer, very potent, you have to go into in vivo, and because it gives more intact biological microenvironment. And so only in vitro experiment, when you are dealing with a, let's say, big shot, big journal, people may not believe what you claim because your observation is limited. Even in vivo, it's more intact environment and it's more relevant to the real human. Even, we can even cure mouse tumor very well, but it's hard to cure human patients. So there's also a big difference between mice and human scale. So when you are dealing with the reviewers, what they will say, hey, are you talking about new cancer treatment and new tumor model? Hey, one model is not enough. For in vitro, they will say, hey, you're talking about breast cancer treatment, and you're bringing only 2 cancer cell lines. Hey, forget about it. You should bring like multiple to show it's more convincing. In vivo, even in vivo, the one model you show that it's effective, hey, that may not be enough. You may want to develop a multiple model and to show that it's more likely to work in human. So that's some of the aspects when you consider, when you have a hypothesis, a new drug, a new method of treating disease such as cancer, and you want to consider where to start. Of course, you probably want to start in vitro. And then once you find the very interesting mechanisms and tendency or potential, and you move on to in vivo experiment. Okay, so let me talk about the final topic, which is systems biology. What is systems biology? It's a study of interactions among the components of biological system and how this gives rise to function and behavior of the whole system. So I would start with this figure. So life science. So, biomedical research usually starts from hypothesis. So this is where you apply for graduate student fellowship, postdoctoral fellowship. Even when you become a faculty, your grant many times required, what's your hypothesis, okay? Hypothesis is a worldview that is testable. So it has to be testable to give a yes or no, okay? Especially in life science, let's say you are interested in the past, one gene at a time. So I'm interested in this gene A, which may be causing cancer, let's say. So that's your hypothesis. Then how do you design your experiment? Hey, I believe somehow this gene A is important for causing cancer. Then you may target this gene and to remove the, let's say, you give this gene A more and more, okay, by genetic modification. So the cancers, the normal cell, you insert gene A, multiple gene A, and then see if they grow like tumor. Oh, yes, that genetic modification. Or the cancer cell, that gene A, you eliminate the gene A and see if this becoming less potent, less malignant. So that's how in the past, our biology, life science has done one gene at a time. But we have a genome and we have over 20,000 of protein. And now it's a year of not gene, but genome. So we are dealing with the whole genome. So we need a quantitative measurement. So that becomes a system. So that's so complicated. You need a concept of modeling. You need to analyze and synthesize the results. And this leads us, even biologists needs to deal with a huge amount of data, and that requires this dealing with the information science. So that means we have database and the model tools, and this is so much that now it's not just a simple one, but you need to have a tool to visualize, and that's called systems biology. Compared with the previous reductionist method is you have, let's say, disease or biological system as a whole, but you cut it apart, like single cell, you study the gene one at a time. So this is called reductionist approach. And we believe that this part we understand, when we put together, we probably understand the whole. And this is a good approach, usually how this science has been approached. But now we have dealing with so many of this a vast amount of big data. And hey, maybe we have to go to holistic ways to look at all the part interactions, okay? And so to see this more of a holistic point of view and systems point of view, and that's where this new discipline of systems biology and how this has been possible is advanced array technology, which means we can now measure simultaneously thousands of genes within a cell or a tissue. And with this data, the application of statistics and computer science to this molecular biology or cell biology field is called bioinformatics. So these people would say, hey, now this is omics era. What does this mean? So this is based on organismal, tissue, and whole cell level measurements. And let's say we observed in 2000 and the DNA sequencing has been done. And what that means, it's a blueprint. Now we have to find out not the alphabet, but the words. That meaningful words is called gene. And all the words like, like dictionary, it's like genomics, okay? So, but DNA, you guys all know from DNA to mRNA and then protein, this transcription and translation, because protein is the functional entity which gives out cellular physiological function. So we have to understand not only DNA, but another level too. So we start to talk about protein, not just single protein, all the protein, multiple 100,000. And to study this is called proteomics. Or people say, hey, I want to know about the gene expression. Okay, all our cells have the same DNA sequence, but why we have like about 200 different kind of cells? And that is determined by specific gene expression. And how do we know that? Hey, not only DNA, the transcript of DNA, which is an RNA, okay? And that RNA, this process is called transcription, and the whole set of this is called transcriptome. So we call it as a transcriptomics, to not only know the basic blueprint, but how gene expression works. And especially now it's an emerging that this developed country, the whole world people are suffering from metabolic disease, which is a multitude of, let's say, diabetes, hypertension, when people lives longer, having more Western diet, less exercise, we all are likely to suffer from this aspect. So studying of this metabolite chemical and whole groups and multitude of this is called metabolomics. And also we talk about carbohydrates. So all the carbohydrates we call glycomics, and all the lipids, lipidomics. So whatever there is, you call it blah blah omics. Okay. So one example of this gene expression value from microarray experiment, and you can see that there are many things. And now we are dealing with hundreds and thousands of a gene expression. How do we express is now requires a tool from computer science, engineering, dealing with this data. And one good way is called a heat map. What it means is it becomes red or green, okay? So normalizing this gene expression, and then if it's higher than normal, you give, let's say, a red color, lower than maybe green color, so that you can see it more comprehensively, and you sort it out based on some rules to grouping together to give out these groups have this tendency and this group have this tendency, this group have this tendency, easier and visualizable way. So, okay, so this is, I think this is at the end, and let me finish. So let's summarize. So so this week we talk about what's biomedical engineering. This is an introduction by example, and we find the role of biomedical engineers for the development of reliable, safe, and inexpensive medical technology. And this gives up extending our lifespan, and especially these days, more of a quality of life. So biomedical engineering is applying science, which is pursuit of knowledge, but we want apply this for human good. So mathematics and engineering design principle to improve our human health. And oftentimes BME have specialized sub-disciplines, which we go through today and on Monday. And now we are facing on new challenges in terms of digital. For example, COVID is a new challenge, and that emerging human disease requires us new discoveries and physiology, how COVID affects our brain function, why this affects our lung, and this human health. This understanding provides us biomedical engineers new problems of the future. So it's keep evolving and it's really